Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
In the race to replace fossil-derived plastics, one question keeps coming up: can we turn low-value cassava-based sugars into high-value PHB in a way that is both biologically efficient and process economical at scale? What makes this study by Asensio Galvan and colleagues so compelling is that it shows how a simple change in feeding strategy, guided by genome-scale flux balance analysis, can flip Cupriavidus necator from just growing to aggressively storing carbon as PHB from cassava-based dextrose. Scientific breakthrough. The core innovation is the integration of fed-batch pulse feeding with a curated genome-scale model, ICNH 2025A, to understand and steer PHB accumulation in necator using cassava-derived dextrose as the main carbon source. Rather than treating feeding as a purely empirical knob, the authors explicitly couple experimental fermentations with FBA predictions of carbon flux redirection from growth toward PHB synthesis under different seed-N regimes and feeding times. The mechanistic insight is clear: timing and composition of pulses reshape intracellular nitrogen status and carbon overflow, pushing the metabolism either toward biomass or toward polymer storage, and the model captures that shift with strong correlation to the experimental data. From an industrial perspective, this changes the equation because it turns a black box fed batch into a model-informed process where you can deliberately schedule pulses to maximize polymer, not just OD. Process and engineering perspective. The authors evaluate 3 pulse feeding strategies in a fed batch setup using cassava-based dextrose as carbon and controlled nitrogen co-feeding. In one regime, they deliver 2 pulses containing both carbon and nitrogen at 24 and 48 hours at an overall C-10 ratio of 73. In the second and third regimes, they use a single dextrose-only pulse at either 48 or 60 hours, effectively decoupling carbon supply from nitrogen availability late in the fermentation. The engineering question is deceptively simple: do you want to feed at the onset of stationary phase to sustain biomass, or later to starve cells of nitrogen and force PHB storage? Experimentally, they observe that early pulses rich in carbon and nitrogen enhance biomass but suppress PHB accumulation, whereas later, carbon-only pulses under nitrogen-limited conditions favor polymer storage, a classic carbon overflow phenomenon, but now quantified for cassava-based dextrose and supported by genome-scale modeling. Quantitative performance highlights. The numbers tell the story. When 2 pulses containing both carbon and nitrogen are added at 24 and 48 hours, seed-N ratio 73, PHB accumulation reaches 33.55% of cell dry weight. A single dextrose-only pulse at 48 hours yields lower polymer, 25.12%, WW, highlighting that simply adding carbon at the onset of stationary phase still sustains some growth and does not strongly push cells into deep storage mode. However, delaying that same dextrose-only pulse to 60 hours increases PHB accumulation to 50.4%, WW, a substantial improvement that clearly reflects a stronger nitrogen limitation at the time of carbon addition. The authors explicitly state that feeding at the onset of stationary phase enhanced biomass growth but reduced PHB storage, whereas late feeding favored polymer accumulation, likely due to nitrogen limitation. An FBA with ICNH 2025A showed strong correlation with these experimental trends by accurately predicting growth and carbon flux redirection toward PHB synthesis. The study does not explicitly report volumetric PHB productivity, maximum CDW, or detailed kinetic parameters in the summary excerpt, but the reported mass fractions already indicate a meaningful move into industrially interesting PHB levels for a cassava-based process. Industrial and commercial relevance. From a process engineering standpoint, the integration of pulse feeding and FBA offers a pragmatic route to intensify PHB production from cassava while keeping the bioreactor configuration relatively simple: no exotic control hardware, just smarter timing and composition of feeds. Feeding strategy is a primary lever on both CAPEX and OPEX in fed batch. Poor timing can waste substrate on non-productive growth phases, increase aeration and cooling demand, and leave you with lower polymer fraction, thereby inflating downstream costs per kilogram of PHB. By showing that a single, well-timed carbon pulse at 60 hours pushes PHB to about 50% of cell mass, the authors effectively demonstrate a way to reduce biomass handling and downstream burden, since higher intracellular polymer fraction typically translates into lower extraction volume and potentially simpler solid-liquid separations. Using cassava-based dextrose as the carbon source is industrially relevant for regions where cassava is abundant and maize or sugarcane are either more expensive or politically constrained. However, the paper does not explicitly present a full techno-economic analysis or compare cost per kilogram PHB against glucose-based processes. For GMP or regulatory contexts, the study is positioned as a production and modeling paper rather than a quality or regulatory treatise. It does not explicitly address impurity profiles, residual substrates, or endotoxin levels, but the Cupriavidus platform is already well studied for biopolymer production, which eases eventual regulatory translation for materials used in medical devices or packaging. Market and business context. PHB sits within the broader polyhydroxyalkanoate (PHA) family, a bioplastic segment that external analyses place in the low hundreds of millions of dollars range today, with projected COGS often quoted in the mid-teens as brands and regulators push toward biodegradable and bio-based materials. The paper itself does not provide explicit market size figures, but by focusing on cassava-based substrates, it implicitly targets markets in cassava-producing regions where there is both a feedstock advantage and a growing pressure to valorize cassava processing residues and avoid waste. Cassava production exceeds 300 million tons per year globally, and its agro-industrial co-products, bagasse, peels, and liquid effluents, represent a substantial, underutilized carbon stream that could feed PHB bioprocesses, further improving sustainability narratives and feedstock economics. PHB competes with other biodegradable polymers such as PLA and fossil-derived plastics. Its key differentiators are biocompatibility, true biodegradability in a wider range of environments, and the potential to derive it from non-food or low-value starch streams like cassava residues. From an investor lens, any process that can move PHB content reliably above 50% of cell dry weight on low-cost feedstocks, while being guided by predictive models rather than trial and error, moves closer to the cost and reliability thresholds needed for large-scale adoption. Startup and investment insight. What makes this truly interesting for founders is that the paper is less about a one-off strain trick and more about a reusable blueprint. Couple simple fed-batch pulse strategies with genome-scale FBA to tune when and how your process switches from growth to biopolymer storage. A natural commercialization pathway is at Cassava to PHB, a platform aimed at regions in Africa, Asia, and Latin America where cassava is plentiful. Here, a startup could own both the fermentation know-how and the digital layer, FBA-driven feeding strategies tuned to local cassava syrup qualities and plant configurations. There is clear IP space around specific pulse feeding schedules, seed-end ratios, and model-guided operating windows for seed necator and related strains on cassava-based dextrose that achieve defined PHB fractions and productivities. While the article itself does not discuss patents, those process conditions and in silico, in vivo integration schemes are strong candidates for protection. From an investment perspective, integrating FBA into routine operations is a signal of maturity. It suggests a process that can be digitally twinned, optimized, and transferred, which in turn supports multi-site deployment and scale-out rather than purely heroic scale-up. The key risks lie in feedstock variability, potential inhibitors in cassava hydrolysates, e.g., residual cyanogens, robustness of seed necator under real industrial conditions, and the capex required for downstream PHB recovery, none of which are fully detailed in this study, but all of which represent opportunities for a technically strong startup to build a defensible moat in process integration and local supply chain engineering. Closing perspective. Stepping back, this work is a textbook example of how modern bioprocess design should look. Start with a real, regionally relevant feedstock, use genome-scale models to understand metabolic trade-offs, and then engineer your feeding strategy to drive carbon into product rather than biomass. By demonstrating that a single, late dextrose pulse can push PHB to around half of cell dry weight in C and necator grown on cassava-based sugars, and that a curated FBA model can predict that shift, Asensio Galvan and co-authors give both researchers and practitioners a concrete, model-guided lever for PHB intensification. For scientists, it's a call to more tightly couple FBA and fermentation. For entrepreneurs and investors, it's a reminder that the real breakthroughs in the bioeconomy may come less from new chassis and more from better orchestration of carbon, nitrogen, and time. The study does not explicitly deliver full techno-economic models or LCA, but it provides the mechanistic and process insights that those models need, making it an excellent foundation for anyone looking to build the next generation of cassava-based bioplastic platforms. For a follow-up deep dive, would you find it more valuable to focus on scale-up and downstream options for PHB from C. necator, or on how to generalize this FBA-guided pulse feeding strategy to other polymers and host organisms?
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Ernala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.